This is our final presentation on the sequence for intracellular membranes, cellular organelles, and vesicular traffic. Now, let's kind of review some things we've talked about. We have talked about constitutive secretion, and that would be material coming from the ER through the Golgi, vesicle pinching off, and the vesicle fusing with the plasma membrane, and the cargo being excreted. And that does not require any special signal. And so I think of examples like that. Many of the extracellular matrix proteins, things like collagens, for example, or fibronectins, things like that, or plasma proteins that the hepatocytes synthesize that are in the blood serum. Those are mostly secreted by constitutive secretion. We also think about regulated secretion, and we can think about that as signal-mediated secretion, where something is budding off of the Golgi and it's forming into a secretory vesicle, and that vesicle stays stored in the cytoplasm until the cell receives some input for secretion. I might think there of something like zymogen granules, digestive enzymes that are synthesized by pancreatic acinar cells, and the digestive enzymes are held within secretory vesicles in the acinar cell, and the cell doesn't exocytose until the cells are receiving some type of a stimulus, either a nerve input or a hormonal input to release the vesicles. So that would be signal-mediated secretion, a regulated secretion. We also think of signal-mediated secretion where material coming through from the ER to the Golgi is diverted into lysosomes. And that's a specific type of signal-mediated secretion, and we know that that secretion involves mannose-6-phosphate receptors. We'll talk about that in just a couple of seconds. To recap what we said, this constitutive secretion, vesicles are pinching off of their donor compartment by virtue of being coated with COP2 proteins. The regulated secretion, vesicles are pinching off from their donor compartment by virtue of being coated with clathrin. Same thing for lysosomes. Diversion to lysosomes with a specific signal. The vesicle is pinching off because it's coated with clathrin. We know that proteins that are tagged with mannose-6-phosphate are destined to go to lysosomes through clathrin-coated vesicles. This signal-mediated secretion is also with clathrin-coated vesicles, so the zymogen granules in the acinar cell that I just mentioned, clathrin-coated vesicles. As I just said, also constitutive secretion with the COP2 coated vesicles. Let's spend a couple of minutes and talk about lysosomes. Now, first of all, what are lysosomes, or how do they appear? With the electron microscope, lysosomes appear as heterogeneous electron-dense bodies, very heterogeneous and very electron dense. A cytochemical stain for lysosomes is acid phosphatase, and as a matter of fact, acid phosphatase can be considered a marker enzyme for lysosomes. So if you stain cells with acid phosphatase and a vesicle stains with acid phosphatase, you are operationally identifying a lysosome. That's the concept of a marker enzyme. A marker enzyme is something that is uniquely highlighting a particular enzyme in a particular organelle. This is the classic pathway for lysosome formation. You have the ER, you have vesicles that bud from the ER and go to the ER Golgi intermediate compartment or the cis-Golgi network. The vesicles move through the Golgi, they go to the trans-Golgi network and bud off of the trans-Golgi network and become what are called primary lysosomes. A primary lysosome is a vesicle that's loaded with acid phosphatase, but there's no intracellular digestion going on. So it's, if you want, a primary or a naive lysosome, as opposed to a secondary lysosome. A secondary lysosome is a vesicle that has acid phosphatase in it, and by which another vesicle has fused with it, so that the cargo delivered to this secondary lysosome from this vesicle is now being digested. And we can contrast that with what's called the residual body. A residual body is a membrane-bound vesicle under which there is no more intracellular digestion going on. We will see the term in the histology laboratory lipofuction granule or lipofuction granules. I've heard it pronounced either way. These granules sometimes are given the term aging pigment. They result as material is broken down in cells and kept within membrane-bound vesicles, but the material is modified and broken down so much that no more intracellular digestion can occur. These residual bodies just accumulate as an organism ages. So lipofuction granules are very common in cells of tissues that live a long time. So for example, neurons have a lot of lipofuction granules. Cardiac muscle cells have a lot of lipofuction granules because these are cells that live almost as long as the organism lives. We can talk about 3 different pathways for lysosomal digestion or lysosomal degradation, and none of this is new to you. You know, for example, that a cell like a neutrophil can phagocytose a bacterium. The bacterium can be trapped in a membrane-bound vesicle. This can fuse with a lysosome and the bacterium is broken down. That's called phagocytosis. Everybody knows that. You also know that vesicles can pinch off from the plasma membrane as an endocytic vesicle, get into an early endosome. From here, they can route through late endosomes to lysosomes, and you can have digestion. So endocytic digestion. Some people call this, if it's just small vesicles, fluid phase pinocytosis. You also might relate to receptor-mediated endocytosis if it's something being brought in with a specific signal, like ligand binding to LDL. And a point that you may not be aware of, we also can talk about autophagic vacuoles or an autophagosome. Let's say you've got some defunct components of cells. Those defunct components might get wrapped around with an intracellular membrane. Here the cartoon is showing maybe little bits of ER membrane surrounding a non-functioning mitochondria. This thing could fuse with a lysosome and you'd have an autophagic vacuole or an autophagosome. So 3 different ways that things can be digested by way of lysosomes. I'm not going to go over this again. We've already looked at it, but here is the internalization of LDL and the receptor going back to the cell surface and the ligand, the LDL, routing to lysosomes for digestion into free cholesterol. Now, let's talk about transport of lysosomal hydrolases, because this is something that is fairly well understood. Lysosomal hydrolase precursors get modified in the cis-Golgi network. And so what happens, here's the green diagram that's saying this is a lysosomal hydrolase precursor. It has high mannose residues already added to it. In the cis-Golgi network, you add N-acetylglucosamine. As this protein hydrolase precursor is routing through the Golgi, it's uncovering a mannose 6-phosphate signal. So that finally, by the time this precursor gets to the trans-Golgi network, this mannose 6-phosphate signal is going to be recognized by specific receptors to that mannose 6-phosphate, and this vesicle is going to bud off with clathrin coats. Now, this is going to be routed to endosomes. As it's routed, it's going to lose the clathrin coat. And this is an absolute requirement that the vesicle lose the coat before it fuses with the target compartment. So donor compartment, the vesicle in transport, the target compartment. So budding off in a clathrin-coated vesicle, the vesicle loses the clathrin coat, it then fuses. After the vesicle is fused, the hydrolase begins to be active because there'll be a relatively lower pH. We know, for example, in these early endosome compartments or early compartments that will become lysosomes, there are hydrogen ion pumps that require ATP that are driving hydrogen ions. We enter the vesicle against a concentration gradient, making the vesicle lumen somewhat acidic. At this acidic pH, the lysosomal hydrolase precursor dissociates from the receptor, phosphate is removed, and you getting an active lysosomal hydrolase precursor, which is going to route to a lysosome. At the same time, the mannose-6-phosphate receptor is going to bud off with a special type of COP1 protein called a retromer protein. It's going to bud off with this retromer coat. It's going to lose the retromer coat as it goes back to fuse with the trans-Golgi network. So it's kind of an interesting way of understanding how the lysosomal precursor gets routed from the ER through to the Golgi. This retromer assembles on endosomes only under highly specific conditions, and I'm not going to ask you to really remember that. You don't even have to really remember that it's a retromer for all that matter. This is what a lysosome is. It's basically a membrane bag that has hydrogen ion pump in it so that it's pumping hydrogen ions against a concentration gradient. So the luminal pH of this vesicle is very acidic, and that's where these enzymes have their optimal activity for degradation, at an acidic pH. Now, there are a whole class of lysosomal storage diseases where there are mutations in the lysosomal proteins, either how they function or how they route to lysosomes. This is clinically really very significant. There's a database called the Online Mendelian Inheritance in Man. You'll actually learn a little bit about that next term in genetics. But it lists any type of clinical disease. If there's a genetic component, it's listed in this. Well, this OMIM lists more than 50 different lysosomal storage diseases that have some type of a genetic component. The genetic component usually is an autosomal recessive disease. One that you've heard of that we'll talk about next semester is Tay-Sachs disease. That's a lysosomal storage disease. It's the most common lysosomal storage disease. We'll talk about that in a little more detail next term in genetics. Most lysosomal storage diseases are due to defects in either the modification or transport of lysosomal enzymes through the Golgi apparatus. Eye cell disease is a relatively rare form of a lysosomal storage disease, and it's also a relatively severe form of a lysosomal storage disease. What happens, the lysosomes lose function, so everything that's got to be digested gets packed into a vesicles. These vesicles get bigger and bigger and bigger, but no digestion is occurring. So it's kind of like having granules that are taking over the entire cell. What happens because of a mis-sorting or routing of the lysosomal hydrolases, rather than them going to the lysosomes, they're excreted from the cell by constitutive secretion. So the cell has no digestive enzymes in its lysosomes. All the enzymes have been secreted to the extracellular space. Think about that for a minute. It's not such a good thing to have digestive enzymes secreted from the cell. The only thing that is a relatively saving grace there. The digestive enzymes in lysosomes are generally active at an acidic pH, and assuming that all other things are physiologically normal, the pH in connective tissue, of plasma, of serum, is relatively neutral, whereas these lysosomal hydrolases are going to be active at much lower pHs. So the issue is not so much that the enzymes are secreted outside the cell and the damage they're doing. The issue is because they're secreted outside the cell, the lysosomes don't get the enzymes, and so the lysosomes can't digest what they should digest, and the cells just become engorged with this material that can't be digested. Let's summarize. The Golgi directs traffic for constitutive secretion or regulated secretion. These are in the exocytic pathway. So constitutive secretion, vesicles pinching off, COP2 coated vesicles, lose the COP2, fuse with the plasma membrane, no signal, the protein is just secreted kind of on a normal basis. Regulated secretion, vesicles pinch off, are coated with clathrin, the vesicles just stay in the cytosol until there is a signal, maybe a hormone or a nerve impulse that signals the cell to exocytose these granules. Regulated secretion. These granules are sometimes given the name zymogen granules. You may recall from biochemistry that a zymogen is an inactive form of an enzyme. Often what happens as these zymogen granules are accumulating, water is being drawn out of the vesicle and the enzymes are getting more and more concentrated and perhaps getting to the point of almost being active. So here's the zymogen granule, the granule pinching off of the trans-Golgi network with a clathrin coat. So the zymogen granule are these immature secretory granules. They're released, as I said, once the cell receives a signal for exocytosis. Now, let me summarize endosomes. We've danced around the issue of what endosomes are, but we haven't really said too much concrete about them. If we reflect for a second and suggest to you that the Golgi apparatus functions to direct traffic within the cell to route things either for secretion or to lysosomes. So the Golgi is a traffic director, if you want, for exocytosis. We can view endosomes as a traffic director for the endocytic pathways. So we can talk, for example, about an apical early endosome or a basolateral early endosome, and what we're suggesting there is you have a vesicle pinching off, going to this early endosome, and maybe part of the vesicle is going to recycle to the plasma membrane, either at the apical membrane or pinching off from the basolateral membrane and going to recycle to the basolateral membrane. But then these early endosomes are going to route material to the late endosome, which is then going to route material to lysosomes for digestion. So you sometimes see in the literature, in textbooks, things that are called multifascicular bodies. They're membrane-bound vesicles that you see with the electron microscope that may or may not have electron density to them, but it looks like they have a lot of different inclusions in them and substances, and those multifascicular bodies would be the early endosomes and potentially the late endosomes. So you can say you've got an apical early endosome or a basal lateral early endosome. So endosomes are directing traffic for the endocytic pathway, whereas the Golgi is directing traffic for the biosynthetic or exocytic pathways. Now, recycling endosomes play an important intracellular storage role for certain types of cells. They will allow specialized plasma membrane proteins to be stored, but then to be rapidly inserted into the plasma membrane under appropriate conditions. This cartoon, let's just say this cartoon is a muscle cell or a fat cell, so an adipocyte or a myocyte. You know that these cells utilize glucose, and you should also remember from biochemistry that glucose is not soluble in the plasma membrane. Rather, glucose crosses the plasma membrane by facilitated diffusion because there are glucose transporters in the membrane. There are also high-efficiency glucose transporters that are stored in recycling endosomes. These high-efficiency transporters will be inserted into the plasma membrane under the influence of insulin binding to an insulin receptor, which initiates a signaling cascade that ultimately causes vesicles containing high-efficiency transporters to bud from the endosomes and fuse with the plasma membrane. And that's exactly what a skeletal muscle cell or a fat cell would need if it needs to take up a lot of glucose. Let me finish this discussion with just a very brief overview of peroxisomes. This is characteristic electron microscopic view of a peroxisome. As you know, peroxisomes sequester hydrogen peroxide, H2O2. So some carbohydrate group, some reactive group, in the presence of oxygen generates H2O2. Now, hydrogen peroxide is a very strong oxidizing agent. It's not good for cells, so cells want to try to sequester the hydrogen peroxide in a membrane bound vesicle. A little bit of an aside, peroxisomes have this very characteristic electron dense crystalline core. So that sometimes when you see that helps you identify the organelle as a peroxisome. So as we said, peroxisome sequesters H2O2. If any of you have had this in biochemistry or maybe microbiology, a marker enzyme for peroxisomes is catalase, because catalase is involved in the metabolism of hydrogen peroxide. So peroxisomes sequester hydrogen peroxide. Peroxisomes, as you might remember from biochemistry, are involved in oxidation of fatty acids. They're involved in metabolism of alcohols, particularly ethanol, and they have this unique crystalloid morphology. This is going to end this last podcast on our intracellular membranes. I want to encourage you, you should be looking at hepatocytes, and you should be looking at pancreatic acinar cells. There is a specific podcast on iTunes that will help you review what the hepatocyte is, and there's a specific podcast on iTunes that will help you review something about the pancreatic acinar cell.